Welcome student. 11th Science Biology Part 2nd. Our current chapter is Human Nutrition. This is second lecture of Human Nutrition. Subtopic is Histological structure of alimentary canal. Gastrointestinal tract is lined by four basic layers from inside to outside, namely serosa, muscularis, submucosa and mucosa. Start from serosa, muscularis, submucosa and mucosa. We have studied this layer one by one in detail. Serosa, it is the outermost layer. It is made up of the layer of squamous epithelium called mesothelium and inner layer of connective tissue. Concentrate on figure C layer the serosa. After the serosa start second layer is muscularis. Smooth muscle form this layer. Muscle arranged three concentric layer: outer longitudinal, middle circular and inner oblique muscles. These three types of muscle form layer called muscularis. Muscularis layer is wider in stomach and comparatively thin in intestine. Inner oblique muscle layer absent in the intestine. After the muscularis, the next layer start called submucosa. It is formed of loose connective tissue containing blood vessels, lymph vessels and nerve vessels. Duodenal submucosa shows presence of glands. After the serosa start muscularis, then again start submucosa and last layer is called as mucosa. In the mucosa, the lumen is lined by mucosa, hence called mucosa. Presence of mucus secreting goblet cells lubricates the lumen of alimentary canal. This layer show modification in different regions of alimentary canal. Modification in different regions in stomach called rouge, gastric gland that secretes gastric juice, small intestine present villi having intestinal gland lined by a brush border or epithelial cells having microvilli. Villi are supplied with a network of capillaries and lymph vessels or lacteals. Mucosa forms crypts in between the bases of villi in the intestine called crypts of Lieberkühn. I repeat, this four layer in histological study of alimentary canal: serosa, muscularis, submucosa, mucosa are four layers. This is histology of alimentary canal. Next subtopic is digestive glands. The glands which present in the alimentary canal except salivary gland. There are three digestive glands present in the human digestive system. The first gland is called as a salivary gland. Another largest gland in the body called liver, and beneath the liver the pancreas is a third digestive gland present in the near alimentary canal. These three digestive glands, then salivary gland, liver and pancreas, we are study one by one. First is a digestive gland called salivary gland. Salivary gland. There are three pairs of salivary gland which opens in buccal cavity. Parotid glands, submandibular gland and sublingual glands. These three major part of salivary glands. Parotid glands present in front of ear. Submandibular glands found below the lower jaw, and sublingual gland present below the tongue. These salivary glands scatter in the buccal cavity. Salivary gland made up of two types of cells, serous cells and mucous cells. These two cells form salivary glands. Serous cell secretes a digestive enzyme called salivary amylase or called also ptyalin. Mucous cell. Mucous cell produce mucus that lubricates food and helps swallowing. Salivary glands working start in buccal cavity when we are ingest the food, and the major role of salivary gland, it mixes the saliva in the food for makes food substance slippery and mix the mucus as well as digestive enzyme in the mouth. Actual digestion start in mouth because the biochemically mucus acts on this food, helps mix with the saliva and makes bolus slippery. Digestion process start from mouth with the help of mechanical activity and biochemical activity with the help of salivary glands. The second digestive gland is liver. Liver is the largest gland of body. Liver. Concentrate the figure. The liver is the largest gland of body. This is vital organ. The color of liver is dark reddish brown in color, about 1.2 to 1.5 kg in an adult. Liver found just below the diaphragm in a right upper portion of abdominal cavity is a position of liver. Each lobe of this bilobe, thin covering called Gleason's capsule made up of connective tissue on the liver. Hepatic lobules, structure and functional units of liver in each lobe. Polygonal in shape, triangular portal area is present at the junction in adjacent lobule in which three is branch of hepatic artery, hepatic portal vein and bile duct. Hepatic lobule consists of cord of hepatic cells arranged around the central vein. In between the cords, hepatic cells sinusoids are seen. The blood flow through sinusoids. Phagocytic Kupffer cells are present in blood. They destroy toxic substance, dead and worn out blood cells and microorganisms. Hepatic cells are produce bile juice, collected and carried through bile ducts and stored in gallbladder. The duct of the gallbladder and hepatic duct together form common bile duct. These are descriptive points of liver. This is structure of this structure show liver and pancreas. Next, function of liver. Stores excess of glucose in the form of glycogen. Deamination of excess amino acids to ammonia. Conversion of ammonia to urea. Synthesis of vitamin A, D, K and B12. Production of prothrombin and fibrinogen is a clotting factor present in the blood. The production present in the liver. Hemopoietic organ. During development, the hemopoiesis activity start in a liver, hence called as a hemopoietic organ, means formation of RBCs, red blood cells. Detoxification process and destruction of old RBCs by Kupffer cells. Secretion of bile juice, which makes food alkaline and brings about emulsification of fats. These are function of liver. Next and third principal digestive gland is a pancreas. Pancreas are leaf-shaped heterocrine gland present in the gap between by band of duodenum below the stomach. It is exocrine as well as endocrine. The pancreas present gap formed by a band of duodenum below the stomach. It is exocrine as well as endocrine. Exocrine part. Exocrine means the secretion take place inside the bloodstream. Outside there is no relation in the bloodstream and secretion. This gland is called as an exocrine gland. Exo means outside, crine means secretion. The secretion take place outside called exocrine gland. In the pancreas, exocrine part, acinar cells secretes alkaline pancreatic juice with digestive enzyme. Pancreatic duct carries pancreatic juice to duodenum. Hepatopancreatic duct is formed by union of the common bile duct and pancreatic duct, which opens into duodenum. Sphincter of Oddi guards the opening of hepatopancreatic duct. After the exocrine part, we are study endocrine part. Endocrine, the secretion take place inside the bloodstream called endocrine. We are study the pancreas glands endocrine part. Islets of Langerhans. These detect the scientist Langerhans denotes the islets of Langerhans. This part present between the acini. They have three types of cell in the islets of Langerhans. They form three types of cell: alpha, beta, and gamma. Alpha cells. Alpha cell secretes glucagon, then beta cell secretes insulin. Insulin inhibits glucagon and controlling the blood sugar level in the body. Delta cells secretes somatization hormone, inhibits glucagon and insulin secretion. Pancreatitis. In the pancreas digestive gland, some causes of this gland, this causes creates a disease called pancreatitis. So what is pancreatitis? Pancreatitis is the inflammation in the pancreas. It's called pancreatitis. So what is pancreatitis? What is the causes of pancreatitis? Inflammation in the pancreas called pancreatitis. To drink alcoholism continuous year to year, and chronic gallstone in the gallbladder, high level of calcium and fats level higher in blood. These are causes of pancreatitis. There are three types of digestive gland: salivary gland, liver, then function of liver, then second salivary gland is third largest salivary gland is a pancreas. Salivary gland, liver and pancreas are principal digestive gland. We are completed two subtopics in this lecture: histological structure of alimentary canal and three digestive gland. We are studied salivary gland, liver and pancreas. Thank you.